The following podcast is intended for informational and educational purposes only. Please speak to health professionals before making any medical decisions.
Gene therapy research can feel like a complex topic, and one that yields many questions. Why are scientists researching gene therapies? How are they designed to work? What are the unknowns? Let us answer some of your questions here, and it's in the Genes podcast. We're excited to feature experts in the field, from research scientists and healthcare providers to an actual gene therapy trial participant. It's in the Genes is produced by Bloodstream Media and exclusively sponsored by BioMarin Pharmaceutical. We're here to turn complexity into clarity. Be sure to subscribe to It's in the Genes from bloodstreammedia.com. If you're hungry for even more on gene therapy research, visit hemedifferently.com and sign up to stay in the know on the latest in gene therapy research. The research behind gene therapy is a new science based around the idea that understanding our genetic makeup, the code that makes us human, could potentially provide treatment for people with rare and genetic conditions. The task of this perhaps seems Herculean and only to be achieved in the distant future. But here we are, with an explosion of gene therapy research programs being pursued for diseases caused by genetic conditions. In our previous episode, we explored the history of gene therapy research and heard stories from scientists on the front lines. In this episode of It's in the Genes, we'll dive into how the research is being conducted, what investigational challenges they have run into, and how are folks accounting for safety? What have the setbacks been, and how have those committed to this science overcome them? Let's discover how gene therapy bumps, stumbles, and sticking to safety contributed to the story.
You can't build roads throughout the country if you don't know what the map of the country is.
That was Dr. Theodore Friedmann, a scientist on the forefront of gene therapy technology from the University of San Diego. So far in our story, the complexity of the science and the mystery of our genetic map happened in a research lab. Dedicated scientists, students, and researchers spent hours, years, and decades unlocking the mystery of our genetic code. Could it possibly be the key to treating rare, chronic, and genetic diseases? Could an idea, this idea, become a treatment option? Research initiatives started developing in several ways across the country. The idea that gene modification could potentially treat rare genetic disorders started in the 1960s and 70s, and individual researchers would hypothesize ideas and perform experiments in private and public labs. In 1990, the National Institute of Health announced that it would form a groundbreaking initiative called the Human Genome Project. The Human Genome Project was created to discover every gene in the human body, what its function was, and to develop a map of that genetic code. The project would be conducted in 5 years and would set out to sequence the entire human genome.
If you're an explorer, you need a map of where you're going to go, and there was very little understanding of the total structure of the human genome, other than for some sequencing work. But with new technology, with machines, with the ability to order bits and pieces of genes with the information technology and tools, computerized tools to put order in it, it became obviously possible that it would be possible to sequence the entire human genome. And that was a technological great triumph. And obviously it's paying off in many ways, but it was attacked early by some people as just sort of science without a question. What are you asking? What you're asking is it's cartography at first, building a map, knowing what the structure is of the material that you're trying to understand.
The Human Genome Project was an important component of gene modification science in that it set out to label each gene and its function. As the project unfolded, NIH Director Dr. Francis Collins held a contest between researchers of how many genes humans have. It was already determined that flies have 14,000 genes, so contest entries for the number of genes in humans were huge. 100,000 genes to 1 million even. Turns out, humans have an estimated 20,000 to 25,000 genes. Not much more than a fly. The work of the NIH and the Human Genome Project was broad and built to assist researchers in their quest for treatments that could fix or replace a non-functional gene. Which brings us to the individual laboratory. How do science experiments turn into treatment for patients?
Well, this is a practice that's been in place for a long time for the many drugs that get developed and tested in humans for either cancer or depression or whatever, and it's referred to as a IND, an investigational new drug, and it has a very specific procedure that one has to follow. And so what one does as a researcher is fairly straightforward, and I'm going to just make up a scenario. Let's say that I want to make a drug for hemophiliacs. Well, first of all, I have my gene that I know is required to make the protein to keep someone from bleeding. And so the patients that I'm going to treat have the light bulbs out, so we know they carry a mutation. And now I want to try to determine, can I put it back into the cell efficiently so that it will work? And typically, you turn at the laboratory, you turn towards experimental settings that let you test that. So you can put it in cells using the virus to deliver the payload. And if the protein is secreted out, you can measure it and say, hey, look, the virus delivered it, the cells are secreting it, and this is what I needed to see in order to go to the next step. So say, for example, if I put 100 trillion billion little FedEx trucks into your body through your bloodstream to try to correct the genetic disease you're carrying, well, I would like to know, where do all of they go? Where did they go over time? So I would test your blood to see how long is it circulating. I would test your urine to see if you're voiding it out into the environment. And I would test your, tears or whatever, throat swabs, whatever, just to see if it's at places where we didn't expect it. And then based on that, we would turn around and say, it looked like you're shedding virus for 2 days, so we want to keep you in the hospital in a contained setting to make sure that there's no risk to anyone else. And so things like that have been done first in animals and then, like I said, morphed into experiments to be done in humans. And with the guidance of the FDA, these are still rigorously practiced assays that are done to ensure safety.
Thanks, Dr. Samulski. Let's hear from Colleen Dansereau, Director of Clinical Research Nursing at the Boston Children's Gene Therapy Center, on what the process is like when the IND or investigational new drug is ready to be tested in humans.
I actually am kind of oversee all the moving parts of the gene therapy program at Boston Children's, getting the patient in, working with the coordinators and the nurse and the investigator, the doctors to, identify patients, get them to Boston for treatment, coordinate all of the pieces. I actually, from a high level now, have research nurses and coordinators and a program coordinator that coordinates that activity, but I oversee that, all those pieces, and then treat the patient and then send them back to wherever they came from, be it international or somewhere in the United States. And then in follow-up to that, anybody who has had anything and has received a genetically modified product, the FDA wants to follow them for 15 years. So then we set up a program for how to follow them from a distance with their receiving home physicians or home institutions. We have a very large international center. So our patients come from all over the world, quite honestly. We have had patients that we've had to get here from Australia and send back, Japan, Vietnam, Ukraine, Turkey. We've had to be quite expansive in our thought and in how we've kind of built out our program.
there has to be physicians that are part of the process, hands down, because they know what the physiology and clinical standards need to be in a particular area. They can think about the things. The nursing staff, very, very, very important to really get down to the nuts and bolts of what this is going to look like and how it's going to go.
This is Dr. Jennifer Adair. Dr. Adair is an assistant professor of the Clinical Research Division at Fred Hutch, specializing in gene therapy research.
And so I think to me that kind of summarizes all of the challenges. Like when you have a really great clinical team and you have, some kind of next generation science, even with all of that technology and the length of time and cost, there's also still this inherent risk with engineered virus particles that we don't get to control where they administer the gene therapy into cells. So any patient that you're going to enroll in a trial has to understand lots of different facets of what you're doing, what you know, what you don't know, that there are risks involved, that they have the ultimate decision in whether or not to participate.
If we know much of anything, it is that humans are complicated and science can be complicated. Gene therapy research is revolutionary science, but there must have been some setbacks to this process. Let's listen in to our experts giving us a glimpse of the setbacks that have occurred over the years and how they are accounting for safety.
The other things that impacted the field was realized it was very early. We didn't understand when we were talking about putting genes back what was the risk and what wasn't the risk and the safety concerns. And so we've had the unfortunate complication where using a different delivery system than for a disease where they were trying to put the gene back into the liver, the patient passed away. And as a result of that, it kind of stunned the field into, hey, what are you guys doing? Is this really, do you know what you're doing? And why did this happen? And so that kind of put everything in slow motion and basically held the field up for a block of time, 8 or 10 years, where it was really difficult to get funding.
There have been quite a few barriers and setbacks. I think first and foremost probably is some of the history that I alluded to previously around the turn of the century, where we had some fairly catastrophic and well-publicized failures, like Jesse Gelsinger, like the boy in the bubble, some of these leukemic side effects that happened with early gene therapies. This really turned many clinicians and even some of the regulators and payers and companies really turned them against the idea of inserting or changing new genes into patients. And this was a real challenge, and this required a lot of hard work to help people to understand that our understanding is much better, and we hopefully have learned the lessons to move forward.
And there was obviously a lot of soul searching and a lot of grief, not only for the child, for the boy and his family, but sort of dismay that the field had again gotten to the point where it was lambasted in the scientific press, in the lay press, and again people were saying that this is not a real field and it's being hyped and it's a disaster. There was a palpable gloom on the field. We were, again, way ahead of ourselves, way ahead of the state of the science.
But there was a core group that continued to work on things like AAV and things like that, and the data consistently kept saying, hey, this one doesn't appear to have toxicity, appears to be safe, looks like it's working, the animals that were blind can see, the dogs that couldn't walk are walking. Maybe we can try this and see if it works in patients. So at this point in time, I would say, we continue to go forward with a lot better appreciation and understanding of safety.
These the setbacks are things that happen in science where you don't know everything you want to know, and that's why they're called trials and they're called experiments and not known to be trouble-free therapies.
And so, for me, it was about kind of owning that responsibility and learning something in that moment. And I think the short answer to your question is the challenges are numerous whenever you're pushing boundaries and doing something new. The biggest hurdle is being flexible and adaptable to meet them.
The FDA and everybody's become more and more astute at what to look for, and so now we have a battery of questions that are asked before a patient can ever enter into a trial, and then we have a battery of assays that are done once the patient gets the therapeutic, and then we have a battery of committees that are referred to as safety board committees that the data is presented to, that unrelated to the person who developed the drug and unrelated to the group that's carrying out the experiments, and their sole purpose is to assess whether or not the results you're getting are within the limits of what one would consider safe and okay to continue.
It's not like just stopping a drug. Once that change is made, it's carried on down through all of the new blood-forming cells that are formed. And so, rightly so, this has caused some very deep, long discussions with regulators about the safety, about having to show that these therapies are safe, that they're not going to cause problems to patients in 1 year, 2 years, 10 years, 20 years time. And so the need to be able to follow these patients up for much longer to monitor the safety has been a real challenge. So we faced a lot of challenges around manufacturing, around quality control, being able to understand what are the components of these therapies that are critical to the clinical safety and the clinical efficacy. So there's been a lot of work around that to really understand this whole new process, because this has been a learning activity for everyone concerned.
There are lots of checks and balances that are built into the system.
There is another checks and balances system in the field of gene therapy research: ethics. What are the moral concerns of gene transfer therapies, and who is leading the conversation?
And so there's a factor of self-policing, but from the ethical perspective, we typically turn to the bioethicists, and they typically have a physical role in making sure that things are done and adhered to. And then there's also been much larger structures, like in the American Society of Gene Therapy. There are typically committees and panels that lay out what we would like to say are voluntary guidelines on what we think is acceptable and not acceptable. And then when it gets to push comes to shove, if it has to go to the next step, we support moving it to here's what everybody agrees to. No one will do the following experiment.
That was Dr. Samulski speaking on the ethical side of gene therapy research. Let's hear from Dr. Sven Kili on his thoughts on the bioethics debate.
I think as we get to understand the genetic modification, the genetic control, things like that, naturally there is a desire for people to move more broadly into different types of indications. And so this becomes a really interesting moral area, that should this be something we go into? Should this be something we can develop therapy for? So there's still an enormous unsolved ethical quagmire that we need to get through for a number of things around us. Just because we can do something doesn't necessarily mean we should.
From complicated science to investigational challenges, to accounting for safety, to a global ethics debate, gene therapy research is a massive undertaking. We asked Dr. Samulski what it would take to make gene therapy a reality.
Yeah, from where I sit, it's been fairly realized that this is like climbing Mount Everest. We've had to set up base camp at different places. How to make the vector, how to demonstrate that it will work, how to show safety, how to produce it. at levels that are going to be sufficient to put into your own kid's body, much less, someone else's. So my motto at the lab was, if you're not willing to inject it in yourself or your own kid, don't hand it to me to use in patients. And it kind of sets a different bar for what you're trying to achieve, because realize we're doing drug development and we're trained as researchers because the field didn't exist, and we've had to develop each of these steps on our own with the guidance of the FDA and stuff like that. They were fantastic partners, and that they provided to a new community some of the things that they've been doing for years for the traditional drug developers. Time is an issue. It's always been an issue. Things need to have been done yesterday. Making sure that we don't leave patients behind, for me, is an issue, a tremendous issue. So you almost have, 14 years of moving that rock up the hill to finally get it to a point where it may eventually get tested and approved. And now you would like to say, we know how to do it, so let's throw them up the hill. Let's get them up there quicker. Let's get a conveyor belt that can take it up, so we're not doing all this legwork and that's so tedious.
But it's not as easy as that, is it, Dr. Samulski?
We've been doing this for over 40 years on a nickel and a dime, and you realize the importance of what it took to make this happen, and the people that made it happen that won't benefit from it or be recognized. And there's been a lot of grassroots work to get this field to where it's at, and I think you can never identify and thank all of them enough, but there are tremendous Mother Teresa's out there with respect to the genetic therapy technology.
Next time on It's in the Genes, a gene therapy podcast, we'll look into the clinical trial process and why the resources Dr. Samulski mentioned are so important. It's in the Genes, a gene therapy podcast is written and directed by Amy Board, produced and edited by Greg Holzman, artwork by Christina Newhart, post-production support from Josh Bragg, Rob Bradford, and Theodore Friedmann. It's in the Genes, a gene therapy podcast is produced by Bloodstream Media and presented by BioMarin. Visit hemedifferently.com for more educational information on gene therapy research. Subscribe, rate, and share It's in the Genes, a gene therapy podcast. Referrals from you are the best way we can reach new people. Thanks for listening. My name's Amy Board, and we'll be back next week with episode 3 of It's in the Genes, a gene therapy podcast.